guys, welcome to this week's discussion video. Today I'm actually covering something a little bit different than what was on the discussion worksheet and quiz. And the reason why is because I got a lot of student feedback asking to go over some of these important experiments that were covered in lecture 8. So these all still have to do with DNA replication, but it's a little bit more specifically focused on the actual experiments themselves and how to analyze their results and what their implications are, rather than the actual mechanism of DNA replication itself. I do have a lot of resources for if you still want a broader concept review of how replication works, but also just recommend reviewing Lori's lecture, reviewing the textbook, and reviewing the video that I posted in my discussion slides. Okay, so there's 4 experiments that I wanted to talk about today: Griffith, Avery, Hershey-Chase, and Meselson-Stahl. Now, they kind of go in this order because it's kind of like the layers of knowledge and discovery that happened over time. So the first one I want to talk about is the Griffith experiment, which essentially discovered transformation and how transformation happens. So the basics of the experiment were that, okay, Griffith's group said, we're going to develop these 2 strains of Streptococcus, one which we know is non-virulent to mice, the R strain. It stands for rough strain. You don't really need to know the structural reasons why this particular strain doesn't cause a reaction and the other one does. But just remember, R stands for rough, and that means no effect. The S strain, however, is virulent. S stands for smooth in this case. Again, you don't really need to know why that structure is important here. But so we know that, okay, if we inject a mouse with this non-virulent strain, nothing's going to happen to it. But if we inject it with its virulent strain, it's going to die. So they want to utilize this knowledge to see if somehow mixing dead virulent cells with alive non-virulent cells, if that will still have no effect on the mice, which is what we would expect, or if somehow there's some sort of transference between these two strains that causes the non-virulent cells to become virulent somehow. So we can see this. These were the trials that they decided to run. So they have the rough strain, smooth strain, confirmed that the mouse lives, and then the mouse dies here. And then they did one trial where they heat killed the smooth strain, because ideally, or because we would predict that the heat-killed strain would not be able to induce an effect in the mouse. So we would predict that this mouse would be alive because the virulence can't actually do anything here. We would expect that this mixture of the heat-killed virulent cells and the rough strain, the non-virulent cells, would have no effect. So we would expect the mouse to be living in both of these situations. And we can kind of, and we would expect that from here because it's essentially just combining these 2 trials. So we'd expect there to be not a difference. Okay, so in their actual results, they confirmed that, okay, heat killing does work in terms of knocking out that virulence because all of those virulent cells are now dead. However, when we mix those 2 groups together, we're somehow getting this mouse dying. So why that's important is that even though they don't know the mechanism at this point or really what's going on, they know that there is something that's causing a transfer of these virulence factors to another strain, another type of cell. And this is important because these cells are already dead. So there's something about it, that something is being transferred that there's something in these cells that even when they're dead, when they get transferred in here, are able to confer those virulence factors, which is pretty cool. Eventually we find out that transformation is possible, even among different types of species. And this has a lot of implications, not only for DNA replication's sake, but also just for the tree of life, that we can have this horizontal gene transfer, gene exchange between species and strains and everything, really just combated the traditional sense of reproduction or binary fission is how we pass on our genes. This kind of added a whole new dimension to that. All right, and so now here we have the Avery experiment, which was able to discover that DNA is the thing responsible for carrying the genetic information. And how it's designed is it actually capitalizes on the original results from Griffith's experiments and uses that to further isolate, okay, what exactly is responsible for this transformation? We know that it happens based on Griffith's results, but now we need to figure out, okay, what is the thing that's being transferred? So how they conducted their experiment was they had this batch of virulent bacteria that's been heat killed. And first they did this control just to make sure that when they added the heat-killed virulent bacteria to healthy, non-virulent bacteria, that that would still cause a transformation. And then they were going to do 3 experimental conditions. In one, they were going to add to the virulent cells, the dead virulent cells, an enzyme that will cleave and destroy any RNA. So any RNA particles are going to be totally inactive in that batch. In the next one, they're going to do the same thing, but with a DNA enzyme. So any DNA that's in there is going to be cleaved and not be functional. Third one, same thing except for proteins. So we can see here, this picture. This is our original heat-killed virulent batch. This was our control group where we're ensuring that transformation does actually happen, which we find. They're able to see, okay, we have this test tube and it contains both virulent and non-virulent. So some of the cells were converted and uptook whatever this strain was transferring to them, and some didn't. So that's why there's a mixture of both. And so here's the tube that has the RNase, which is the enzyme that cleaves RNA, proteins, and DNA here. And so what we're kind of expecting to find in this experiment is, okay, we know that if we have these enzymes that destroy a certain component, if that component causes the transformation to stop, that like there is no transformation that's occurring, that component that we kicked out must have been the thing that's responsible for this transformation to begin with. So it's kind of like an elimination trial. So essentially we're just trying to find, okay, which test tube is going to look different than this control, and that will tell us what the genetic particle is. So here's the results. And like as we expected, some of them actually didn't have any effect. So when we cleaved the RNA, that didn't have any effect on what was transferred and how it was able to uptake these virulence factors. Same with the proteins. But suddenly when we stop having this DNA transfer occur, now suddenly they can't uptake these virulence factors anymore. So ultimately we can conclude, okay, this is telling us that DNA must be the thing that's responsible for holding this genetic information. Now, unfortunately, this was not enough to convince a lot of scientists, especially because at the time, the predominating theory was that proteins were responsible. So there's already kind of this resistance, and then just having this one interesting experiment result didn't really do much. So they were like, we need more evidence. And luckily, Hershey-Chase was able to provide that evidence. How they set up their experiment was that they utilized a bunch of information that they already knew about viruses to be able to design this experiment that can kind of eliminate one or the other. So we know at this point that viruses contain both protein and DNA, and those were the 2 main contenders at the time. So they really wanted to just eliminate one of those 2. They know that viruses have to hijack cells in order to copy their genome. They don't have the cell machinery available to copy their own. So whatever they infect other cells with has to be the thing that they're trying to replicate. And so also, another helpful factor is that we know that the protein and DNA are located in separate areas of the virus. So essentially the outer capsid of the virus is going to be that protein coat. It's going to be lots of proteins there. And then the inside of the head is going to be where the DNA is located. So they're able to determine where things are and tag them with different elements, different radioactive elements, in order for us to kind of look at, okay, what is being transferred out of the virus when it's infecting a cell, what's staying in the virus, and that will tell us, okay, where is the genetic information located. So they tagged protein with a sulfur isotope and DNA with a phosphorus isotope, each of these which were radioactive. And essentially they're just trying to find out, okay, what's going to be located in the infected cell after the virus has injected whatever material it's injected. If we see sulfur inside the cell, that will tell us, okay, obviously sulfur, or obviously protein was the thing that it's trying to copy, so that must contain the genetic information. If we see phosphorus inside the cell, that will tell us, okay, that must be what's responsible for holding the genome. So we can look at the results here. Oh, actually, this isn't the results yet, but this is essentially just a graphic depiction of what I just said. But so here we can see this protein coat, this outer layer being tagged with sulfur, and then a cell being infected, and then whatever happens here, if we detect radioactive sulfur here inside the cell, then that's going to tell us, okay, it must be protein. And then opposite here, but we can see that the DNA is located inside this head. Okay, so their actual results, what they found was that there was no radioactivity inside the cell. And what this tells us is like, okay, here's the protein coat, this green layer. And after it's infected, it's all remaining on this actual virus. body and not actually being inserted into the cell. And if the whole point of inserting into a cell is to copy a genome, then clearly the proteins will not actually be holding any genetic information. When they did the DNA trial, they were able to corroborate Avery's results because they were able to see radioactive phosphorus inside the cell afterwards. So they know, okay, that must be what's responsible for holding the genetic information. Okay, so at this point, we have enough evidence to say, okay, DNA is the thing that holds the genome, but we still don't really know any mechanisms. How does that happen? So Meselson-Stahl was able to identify which of the 3 predominating hypotheses at the time were correct. And so if we first just want to review, even before we get into the experiment, just to review what those 3 main models were and what they look like, and then I'll go into the experimental design. So there's 3 different types, these 3 different models. There's the conservative model, semi-conservative model, and dispersive model. Now in the conservative model, we're able to see that the original parent strand acts as kind of a master copy. So when it gets replicated, we have one complete original DNA, and then we have another copy that's completely new DNA. And that happens at each successive step. So if you look at just this being the parent particle, at that next step, this is completely being transferred here, brand new particle here, and same thing here, and so on and so on and so on. Now, in the semi-conservative model, instead of the conservative model where you have one completely old and one completely new, instead it's kind of a middle ground. It's saying, instead, we have one strand from the original parent's helix being transferred to each of the daughters, and then each of those strands is a template for a brand new strand. So here in the first generation, we have these 2 new hybrid molecules, and then at each successive step, that still happens. So we can see, it's pretty easy to see with this dark blue strand, okay, obviously the strand is being transferred here, and then the corresponding light blue strand is gonna be completely new DNA. And then this is harder to tell, but okay, we can see this light blue strand is going here. So one of these light blue strands is from the parent and one of them is new. And and obviously that's happening each time, and for further generations, the same thing would happen, where you get one strand from each, and then the others, all the other strands will be new. All right, and now the dispersive model is a little bit different than both of those 2, in that it's saying that at every step of the way, every replication step is going to result in a hybrid of not only the helix itself, but of the individual strands. So here, we saw there's 2 hybrid molecules because it contains one old and one new. But here, even each strand contains old and new. So every single strand is going to be some sort of hybrid. And this is pretty easy in terms of identifying what's going to happen throughout the generations, because we just know that it's going to be hybrids all the way down. Okay, and so now moving on to the actual experiment part. First of all, I just want to rave about this experiment because I think it is so elegant. It's so simple. And when you read it, it's like, oh yeah, that makes a lot of sense. But the fact that they were able to like come up with this out of nowhere and like so succinctly be able to determine which one of these was correct is just like absolutely mind blowing. So I wish I could like have coffee with them. But anyway, they're essentially deciding, okay, we need to do something that will somehow differentiate old DNA from completely new DNA from hybrid DNA. And so how they decide to do that is with isotopes. And so what they decide to do is utilize differences in weight between isotopes in order to do that. So they decided, okay, we're gonna take our parent cell from a medium that contains only the N15 isotope of nitrogen, which is gonna be a little bit heavier than normal. And that is going to be what's in our parent strand exclusively. And then at every subsequent replication step, we're gonna be conducting this in a medium of exclusively 14N, which is a lighter isotope. If you remember, isotopes are the number of neutrons. So this will have one less neutron than this, which will ultimately add up, looking at entire genomes. And so we know that the heavy isotope is going to be further down in the tube. It's going to be more dense. It's going to weigh more, so it's going to be down here compared to the 14 nitrogen isotope. And the interesting thing about this is that we know, okay, if a daughter cell has 2 strands that only contain this 14N isotope, it's going to show up right here. If it contains one strand of the old, heavier isotope and one strand of the new, it's a hybrid, it's going to be kind of in the middle because it's going to have one of each. But if it has 2 of the heavy strands, it's going to have a band down here, which means that it's going to be from the original medium. Okay, So now we can look at our different models and make predictions on what we would expect to see in this experiment based on if each of the models was correct. So first, looking at the conservative model, which I'll try to zoom in a little bit. Okay, so looking at this conservative model, we need to determine what we would see in this tube after the first generation, after the second generation, and then like what would keep happening after further generations. So after this first step, if you recall, we have this original master key kind of thing, where we have one completely old parent DNA and one completely new DNA. So at this point, we can say, okay, now there's still going to be this band here because we do have this, but there's going to be progressively more at this completely light step. So another interesting feature about the conservative model is that we would expect to never see a line at this hybrid because we're never actually hybridizing between the two different types of strands. Okay. This is just an explanation of that. Okay, for semi-conservative model, we look and we see, okay, after the first generation, we expect to see exactly half from the original, which contains that heavy isotope, and half from the new, which contains the light isotope. So we would expect to see just a single band here. Even though both particles are hybrids, it will just appear as one band because they're both going to be in the same place. And so that's helpful. So we know that we're going to have one band here. And then after this generation, okay, we see, well, each parent is donating one strand to each of the daughter cells. So we can see this dark blue is transferred here, and then light blue transferred here, and then blah, blah, blah. So at this point, we expect to see about a 50-50 mix because each one contains this original DNA being donated to half the daughter cells. So you'd expect to see a line still at the hybrid, but we would also see a band now at the N14 because we have these two strands that were donated the new strand from the previous generation that already contains the lighter isotope, and then obviously any new strand is going to contain the lighter isotope. So we'll have another band here. And one thing I wanted to mention, just because I feel like if you just look at this level of generation, it looks like, oh, it's going to be 50-50. We have to remember that replication is exponential. And the fact that of these 4 particles, there's only 2 of these original dark blue strands and 6 of these lighter blue strands that are each going to be donating to the next generation, and so on and so on. We're going to get a massive buildup of just the nitrogen-14 bands relative to the hybrids, just because that is an exponential increase. So like here we have 100% hybrid, this one we have 50%, the next generation would be 25%, and so on and so on and so on of the hybrids. So we would, if we were to continue this on, we would just see that this band here, this lighter band, would be getting thicker and thicker and thicker. So that's another way that we could detect the semi-conservative model. Okay, and lastly, we have the dispersive model, which states that not only are you going to have this hybridization between a parent strand and a daughter strand, like we see here, but we're going to have hybridization of the actual strands themselves. So even these individual strands are hybridized. And basically what that means is that it's going to be super easy to detect in this experiment because you're essentially just going to have a band at the hybrid after the first generation. And then after the second generation, it's going to just get thicker and thicker and thicker and thicker the longer that you keep it running. So that will be really easy to detect if it's the dispersive model. The one interesting thing though is that after the first generation, you're not going to be able to tell between these two models because they're both going to just appear at the hybrid line. We're not going to be able to detect based on this experiment, is it the strands themselves or is it just that one strand is coming from one place and one strand is coming from another. So now it's time to analyze our results. We can see at generation 0, that's just showing that this is where the parent DNA is coming from. So it's exclusively at this heavy isotope because that's the media that we're taking it from. But now when we switch to the new medium with the lighter isotope to create this first generation, now we see a single band at the hybrid line. And immediately, already, without even looking at what the second step says, we can eliminate one of the models. And you should be able to tell me, okay, the one that we can eliminate is the conservative model. Because if you remember what we said before in our predictions, since we only have this one like master strand, that's just anything that's copied off of it is going to be completely new. We'll never be able to have an instance where we have a hybridized molecule because we're always just going to have an 100% heavy and 100% light. So immediately we can knock that out of the park. The reason why we couldn't eliminate anything else at this step is because these two, semi-conservative model and dispersive model, are going to look the same. at this point. So this isn't going to give us enough information to tell us, okay, is the entire strand being hybridized, like the strand itself, or is it just that one strand is coming from the parent and one is new? We don't have enough information for that yet, which is why we have to do a second generation to further detect what's happening. Okay, so now we look at generation 2 and we say, okay, we went from this hybrid line, and now we still have a line at the hybrid line, but now we have this new band at the N14, the lighter isotope, 14 isotope. And immediately you should be like, okay, we can eliminate dispersive model, because remember we just said it was hybrids all the way down. You're never going to get anything but hybrids for this model. So immediately we can eliminate the dispersive model. But not only that, you can also prove the semi-conservative model based on this. Because if you remember, we said, okay, here, the first step, we're going to have only hybrids. The next, we're going to have about 50-50 hybrids and completely new. So we can see about 50-50. And if we were to continue doing this on, like I mentioned before, we would get this line getting progressively thicker and darker like this compared to this one because of the exponential growth. Sweet, that's pretty much all that I wanted to cover in terms of the different experiments. I know that they can be kind of hard to remember which one is which, but I think it's really helpful to think about it in terms of the order. So if you look back here, going in this order of, okay, what are we discovering at each level? We're getting like more and more information about what's going on. Yeah, so I tried to split these slides up in terms of not revealing the answers on a given page. So I would like wait a little bit, but before the exam, come back to these slides. And before you look at the answer slides again, you should be able to predict, you should be able to say exactly what's going to happen and why that's important, how it's relevant. And that will get you a long way in terms of studying for the exam. So good luck, guys. And also, since I know that this video isn't covering the actual mechanisms of replication as we know it today, the semi-conservative model, if you still want more help with that, please, please, please let me know. I don't want to leave you in the lurch. But again, go through Lori's lecture and the video that I posted, and that hopefully should be a good overview, but please don't hesitate to reach out if needed. All right, guys, thank you.